as you know, from doing it the first time. So you'll do your best to pick however many you decided to pick onto each of the plates. And if you can't get them all, I'm happy to come and help you. So if you wanted to have like 5 worms or something like that, and you can only get 2, I will help you get the other ones so that we can set up your paralysis pathway. Okay, so that's the plan. Let me see. Sorry, it has to stop blinking before it can switch to the laptop. Yeah.
Are we synchronizing the development of the worm?
That's a great question someone was asking. So that's a common way to do it, and it is effective to get a whole population of worms at the same stage. But the bleach synchronization itself is a little tricky. So I just set them up so that you will have the right stage there, and I checked to make sure that we do have the right stage. So we're not gonna do the synchronization step. If you were doing like a longer project, I think it's another skill to learn, but for this, yeah, we don't have a ton of time this summer, so. Okay, so this stuff is lab application, so I kind of also like to, show you how this stuff applies to today's content. Even though the lac operon itself is in bacteria, we use this in biotechnology all the time. And in fact, in our RNAi experiment, we are going to be using the lac system to induce the expression of the double-stranded RNA. So I'm going to teach you about some extra tools that have been developed that kind of work with the lac system. And the first one is called IPTG. So this is what it stands for, isopropyl beta D thiogalactoside. What this mimics is lactose. And you might not think, well, that doesn't even really look like lactose. Part of it does look like lactose, right? Remember that lactose is a disaccharide, it has 2 sugars. So part of this that actually interacts with the repressor resembles the actual lactose substance that normally binds to the repressor. The reason why people use IPTG instead of using lactose is that this is, well, first of all, it's structurally related, so it binds to the repressor. It acts as an inducer, but it's not a substrate. So if we think about it, lactose is the thing that turns on the lac operon, but lactose is also digested by, once you turn on the lac operon, it is a substrate of beta-galactose, right? So not beta-galactose, beta-galactoside, what is the beta-gal? Anyway, I can't think of the actual name of beta-gal right now. So we use it as a stable artificial inducer for the lac operon, and we can add IPTG to any source, right? So we have this as a chemical. When we induce the RNAi, we're going to add IPTG to induce the expression of the double-stranded RNA. And it gives you that control of when do you turn on expression. You can tightly regulate that. This graph right here is showing that this is the lac operon mRNA levels that are being measured, and this is the time after IPTG has been added. So you have very precise control of when you start inducing the expression of the lac operon. Okay, so how do we use the lac operon if we're not really using, we are using bacteria, right? But we're not trying to get them to express lac Z, lac Y, lac A. The way that people have done this is they take just the lac regulatory sequences. So if you think about it, we have the operator, we have the promoter, right? We can just take that part and we can move that to control something else. It doesn't have to control expression of lac Z, lac Y, and lac A. So in the case of a lot of biotechnology, we will put a gene of interest downstream of the lac promoter region, and then that makes it inducible for us. When we want to turn on the gene, we add IPTG, and it starts producing whatever gene we put downstream.
Is that in place of lac Z?
Uh-huh.
Oh, okay.
Yes, so it's kind of like taking this modular piece of DNA and hooking it up to a different gene. In the case of our L4440, we did a very similar thing. Well, they didn't put the lac control region, it's kind of an indirect process. but they have a promoter going in one direction and a promoter going in the other direction to produce, this was our gene of interest in this area, whichever gene you selected. And our goal is to, when we want to turn on expression, we want to turn on from both promoters so that we get one RNA going this way, one RNA going that way, which is going to bind together and form the double-stranded RNA. We're going to set this up, instead of using the lac promoter, kind of indirect, we produce the polymerase that responds to this promoter. And that is what is under control of the Diane. We have the lac promoter, which is controlling the synthesis of the T7 polymerase. Okay, so that's going to make the polymerase that binds to the T7 promoter. So in a way, adding IPTG to turn this on is going to make the polymerase, it's going to then start transcribing. So we'll have the control of when we start making double-stranded RNA. Does that make sense to everybody? It's kind of a little bit complicated. All right. But I want to make a note that we kind of, we'll talk about cases where you want to produce, let's say you want to produce a human protein. If you want to make insulin, for example, and you don't want to harvest it out of another animal, you could put the insulin gene right here, and then put this into this plasmid, and then basically if you put that in E. coli host cells, whenever you add IPTG, you're going to start making insulin, or you could start producing growth hormone, whatever gene you put there, you can start producing it in the bacteria, and then you harvest out that protein out of the bacterial cells. Yeah.
So, to grow the E. coli, then you add the lac promoter so that the T7 polymerase gets produced and gets attached to the L4440 promoters.
Yeah.
Then it goes, okay.
Does that make sense? Yeah. So everything is, like in our system, everything's already in the bacteria. It's just a matter of when we add IPTG, then they'll start making the double-stranded RNA. Okay, so that's kind of the idea of using the promoter sequence. People have also used the LAC-Z gene itself. So this is useful because LAC-Z, oh, that's what that word was what I was trying to think of. Beta-galactosidase. So this LAC-Z gene, of course, encodes beta-galactosidase. We know that that can process lactose, but it also can process other substances. I'm going to show you this substance. This X-gal substance is a chemical substrate that when it gets cleaved, it produces a blue product. So it's basically a readout. If you have beta-galactosidase there, which means that LAC-Z was turned on, you'll get a blue product. And that can tell you when LAC-Z is being expressed. You can use this with cloning. There's different cloning applications where you'll be able to have blue colonies, and white colonies are the ones that did not have LAC-Z being expressed. In the case of systems, we can use this this was taken from a paper where they were trying to figure out where a certain promoter was being turned on. This was a stem cell promoter. So they're basically saying, where are the stem cells in this mouse? When is the stem cell promoter being turned on? They looked at neonatal mice in all these different tissues and adults. The PROM1 is that stem cell specific promoter. Wherever you see the blue color, that's where that that promoter was turning on the expression of LAC-Z. So that's a way for them to kind of visually see where it was turned on. So my point here is that you can take either the promoter from the lac operon and tie it to a random gene, or you can take the LAC-Z gene and tie it to a random promoter. You can use both parts of it. And again, for this stuff, I'm just showing you applications, so you don't need to know the specifics about that stem cell thing. So are there any questions about how this can be used? Yeah. Oh.
Just for clarification, why is the second, like this method beneficial, knowing when the lactate is turned on?
Yes, it's not so much about, this is kind of an artificial setup, because that's not really, LAC-Z is never found in mice anyway, but it's being used as a color reporter, as a visual way that you can then see which cells turned it on. It's kind of like using green fluorescent protein as a visual reporter for some, where a protein is localized or something like that.
So they attach the LAC-Z protein to a promoter in one of these kinds of cells and then just wait it.
Yeah, so they made a mouse that had the stem cell promoter
Oh.
hooked up to that, because they didn't know when or which tissues that stem cell promoter was being turned on. Yeah. Okay, and I'm gonna skip that slide, that's sort of another application. But that's it for the LAC, for this prokaryotic gene regulation. Anybody have any questions, either from your ICA or from any parts of what we talked about yesterday? Okay. So I guess one note, the one thing I will mention is one way that I often put this on a test with the LAC promoter, sometimes I give you a chart that has different situations. I'll give you an example of this. I might have, like, put IPTG, well, let's start with lactose, glucose, IPTG, and then the question might be transcription. And this is basically saying that with the LAC operon, then I would put like plus, plus, minus, right? Or minus, plus, plus. And in each of these situations, you have to think, all right, if lactose is there and glucose is there, there's no IPTG, would I get transcription of the LAC operon? So what do you think for that one? The first one. So we have lactose, but we have glucose. What would be transcription? Plus, 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 or minus?
Plus.
Just one, right? Okay. Then let's say this situation, there's no lactose, there is glucose, there is IPTG. So this one we haven't talked much about, but we can always think of this as doing the same thing as lactose. So it's exactly the same, right? If we had minus, minus, plus, what would you say? Yeah, plus, plus, plus. Okay, so I might give you a question in this form, or I could even make it more of an application question to say, would it be a blue colony? So blue colony, remember, if X gallons there, it's gonna be blue if LAC-Z is turned on. So for this, you could say, all right, well, it's 1 plus, so it might be light blue. Again, light blue. If it's plus, 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 the colony will look blue. If it's a minus, no transcription, the colony would look white, because we're not, the X gal is not being processed to produce the blue product. So sometimes you have to like take what you know and take it one step further. Does that part make sense with X gal? We can do some more review practice problems on that tomorrow. Okay. All right, so now we're gonna switch gears and go to the gene regulation in eukaryotes, which is more complicated, of course, because eukaryotes are a lot more complicated. So we're gonna talk about the added layer of chromatin structure and nuclear architecture, which affects whether or not you can turn on bees. So we've already talked about this, right? These are all the places we discussed for bacteria, that bacteria could control transcription or translation, or post-translational modifications could also control the expression. But which place did we say was the fastest place to control? Yeah, this, right? Because this is fast, we'll just get rid of the protein product, but it's also wasteful. We said that it's more efficient or more energy saving if we start and and regulate the transcription. So for eukaryotes, they can regulate at all these points as well, but in addition to these, they also have additional steps, points of regulation. So they have the added regulation after transcription, which is called post-transcriptional regulation. They can control splice products. We talked about alternative splicing. We talked about all the steps that have to take place, the processing. What were those things that had to happen? Yeah, we had to do the cap, the tail, we had the splice. All of that was required for the export. And all of that actually could, the poly A tail in particular, can affect the stability of the mRNA. So there's lots of levels of controlling even the RNA to say whether it even gets translated. And then additionally, eukaryotes have another place they can regulate, which is how the DNA is packed. So that is called chromatin modification. So we're going to talk about what is chromatin and how does that affect whether or not genes are going to be turned on or not. All right, I think I gave you this DNA packing paradox at one point. But if you were to stretch out your DNA from end to end, anybody remember how long it would be?
2 meters.
2 meters, right? So we did the calculations. We have 3 times 10 to the 9th base pairs per haploid genome. Base pairs times 3.4 angstroms per step per base pair. And that is going to give you 1 meter. It'll give you, I wrote it down, 10 to the 10th angstroms. Okay, which is about 1 meter. That's for the haploid. So since you're diploid, we would double that to get the length. So 2 meters. 2 meters is like 6 feet. So that's really, really long. Does anybody remember the size for the nucleus or just the range? Can you remember the units that it's in? That might be. So it's in micrometers, micrometers, right? So it's about 10 micrometers. And micro is 10 to the minus 6 of a meter. So we're cramming 2 meters worth of DNA into 10 times 10 to the minus 6 meters. So that is a lot of packing that has to happen. So how can we possibly fit that? Especially when we add on that we have negatively charged DNA. It doesn't really want to pack together, right? So the problem is solved by these hierarchical folding of the DNA. So we have to pack it. It starts from here. So this is your naked strand of DNA. The DNA is wrapped around these histone proteins. And then this is actually super coiled around itself to form these solenoids that form a thicker fiber. Those are then looped, and those loops are formed into rosettes, and the rosettes are compressed on each other. So that's a lot of levels of folding. I don't know if any of you have tried when you pack your clothes and you tried the rolling method, and allegedly you can fit more. I still am just a traditional packer. I never try to roll my stuff. But the idea here is that by rolling, you're going to be able to pack more of it in a small space. So the answer to the paradox is through chromatin. Okay, so chromatin we're going to define as the DNA complexed with histones and other proteins. And we'll talk about how exactly that packs together in the next couple of slides. With histones and other proteins. Okay, and there's 2 basic types of chromatin. So there is euchromatin and heterochromatin. This is showing 2 different nuclei. They're actually both immune cells. And you can see that one of the nuclei is very light staining. It doesn't look dense, right? The other one, the outline is kind of around this sort of bloody shape. But this you can see is a lot more dark, it's dense staining. So this nucleus has more heterochromatin than that nucleus. I want to emphasize both of them have both types of chromatin. So most nuclei are going to have sections that are heterochromatin and sections that are euchromatin, what we're comparing here is which one has more heterochromatin compared to that one, right? So the euchromatin is not as dark because it is open. It's sort of decondensed. It's not that tightly packed, rolled. It's mostly in the 10 nanometer form, so that more open format. And functionally, it is more active. So because it's less packed together, I'll go back to this picture. If you think about for transcription to happen, what has to happen in order to be able to express a gene? Which proteins have to get in there and do stuff if you want to have a gene be turned into mRNA? Yeah, RNA polymerase has to go in and find the promoter, right? So you can imagine if you're looking for a promoter and you're looking in the depths of this rosette, it's going to be buried. It's going to be really hard to access. But if you were searching in strands that were open and spread out like this, it would be a lot easier. So transcription happens in the decondensed regions of the genome. So those are the transcriptionally active. The other type, the heterochromatin, the dark staining parts, this is highly condensed. So we're talking about those rosette structures, closed off, inaccessible to like RNA polymerase and other enzymes. And because of that, they're silent. Those genes that are packed away, it's like your clothes that you put away for the summer, pack away all your winter clothes, you're not going to wear them during the summer. So this doesn't get expressed at all. And then within this type, heterochromatin, there's two subtypes. So there is constitutive heterochromatin and facultative. And yesterday we talked about what does constitutive mean? Constant, all the time. So if it's constitutive, it would be always turned off, basically never expressed. We'll talk about some of the places in the genome that are never turned off, they're always silent. Facultative means you can be turned on or off, depending on the environmental conditions. Okay, so the chromatin itself, we said it's DNA complex with histone, so let's look at the basic unit for chromatin. The basic unit is called the nucleosome. And the nucleosome has two parts. It's the protein part and the DNA part. The protein part in this picture is represented by just a single wheel, but it's actually made of 8 histone proteins. And it's essentially an octamer of dimers, which that's how you get 8. So there's 2 histone H2A, 2 histone H2B, 2H3 and 2H4. And they all form, it looks like a wheel. The crystal structure actually shows it to be like the wheel shape. Around that, you have DNA that is wrapped. And this core DNA is a characteristic that's actually conserved across organisms. So there are 147 base pairs that touch this wheel. When we say core DNA, that's what we're talking about. It's the section that is touching the wheel. The intervening stuff, this is called linker DNA. And as you can see, there's a range. So depending on the species, there's different linker lengths. Sometimes they have more space in between, which would mean that it's sort of unpacked more. And sometimes it's that shorter linker length. Okay. Any questions so far on those parts? All right, so when we look at the actual crystal structure, here you can see the wheel hub. Now they have color-coded all the histones. So you have 2 H3s, 2 H4s. Those are brought together on that top half, and then the H2A, H2B are together on the bottom half. And you can see the DNA wrapping around. It's almost 1.5 times it goes around. Overall, the general characteristics about histone, let's just think about it for a second. If we have negatively charged DNA that we're trying to pack together, what would we want to do for the proteins that interact with that? What kinds of charges? Positive, right? So they have a lot of these basic residues, lysines and arginines, that carry a positive charge. And the idea is that it's neutralizing the negative charge so that the DNA can actually pack together tighter. The other thing they have, which you kind of see floating off this wheel, are these tails. And this is really, really important because the tails will determine whether this is packed very tightly in the form of heterochromatin or whether it opens up. So this is the site that you can basically modify to determine if it's going to be closed off or open up. And we'll talk about how that is determined in a couple of slides. Okay, so those are the 4 histone proteins that are part of the central core. There's also other histone proteins that help it to pack even further. So we said that when you go from naked DNA to having these histone proteins, we form the first level of packing, which is 10 nanometer fiber. That's basically the diameter across. And you can see in this picture, it's like beads on a string. That's what the 10 nanometer fiber is, the most open that it can be. If you add one more histone protein called histone H1, this is like a linker, and you can see what it does to the linker DNA. It just tightens it up and compresses it more. So as soon as you add H1, you get a thicker fiber. So this is called the 30 nanometer fiber. And hopefully in the picture you can appreciate that this is just a thicker beads on a string, right? So that's the second level of folding. From there, you can form these loops. And this is actually an electron micrograph showing some of the DNA extensions, the loops. And they think, so this is still kind of a debated thing, that there are proteins that scaffold it. There's always been a debate in science where once you use microscopy to look at something, you've always manipulated it. So, for a long time people were like, well, you basically fried the sample, and so now you think there's proteins down here, but is that actually really occurring? But they do think that there are proteins that hold the bottom of the loop. And as you might imagine, right, if you have genes way down here, they're probably not being expressed. The things that could be accessible have to be, in the 3-dimensional structure, they have to be accessible. All right, so those are kind of the idea behind that loop. All right, so now we've talked about, the feature or kind of the general levels of folding. Now let's actually talk about heterochromatin itself. We said heterochromatin, is that the active or the silent form? Silent, right? So highly packed. Because it is highly packed, it is resistant to basically any enzymes or any proteins. They can't penetrate it. So nuclease is an enzyme that will cut up DNA, but your heterochromatin is not going to be accessible. So that's resistant to nuclease. It's also going to be resistant to any kind of the other proteins like polymerases or even DNA replication. So your heterochromatic regions of the genome, they get replicated last because you have to kind of unpack them a little bit to get access to them. And actually, this is something to think about. When we drew out our DNA replication bubble, did we consider that the DNA is actually wrapped around nucleus? So we left out that detail. It really complicates things. But in actuality, in addition to replicating the DNA strands, you also have to replicate those proteins. So as you're moving through, it's not just like this bubble that we drew like this, because all this DNA was wrapped around nucleosomes. So we're not going to talk about how they do that, but just be aware of the complexity of that. All right, so parts of, on this sheet so far, on your ICA, so far we have answered questions, the first row here, transcriptionally active or silent. So you can fill that in for euchromatin and heterochromatin. So let's take a moment and do that, and then we'll talk about what regions of the genome we find these in. So now going to the second row here, the regions of the genome. For euchromatin, it's basically any areas that are actively expressed. So any of the regions, any genes that are turned on in that cell, they will be found in euchromatin format. And then for the heterochromatin, I have a list here of places in the genome and describing what type of chromatin they have. So we mentioned there are some places that are constitutive, they're always going to be silenced. And one place that we silence all the time is the centromere. So just to remind you, on the chromosome structure, we've talked about telomeres. Where are the telomeres found? Yeah, so you have telomeres way out here. The centromere, as the name kind of suggests, is this region in the middle. And each strand has its own centromere. So if you think about it, this is one strand of DNA, this is another strand. So here there are two centromeres. They usually are found in the middle, but in certain chromosomes they can be shifted, but it's generally found at the sort of the center part of the chromosome. And those are always silenced. The reason is that they have a functional job. In mitosis, they're going to have to hook up to the kinetochore proteins. If you kind of remember learning about that, or even if you don't, we'll talk more about that. But there are proteins that attach to the centromeres, and those proteins attach to the mitotic spindle, to the microtubules. So because they have that role, we're not expressing genes out of them. So they are constitutively heterochromatin. Another place where we have constitutive heterochromatin is in the mammalian X chromosome in female cells. So how many of you learned about X inactivation before? Does anyone kind of want to explain just whatever you remember about it? Do you want to share?
Well, there's two XX, if you're female, you have two XX, and so one of them has to get turned off.
Yep.
And express it.
Yeah, exactly. So that's the dosage compensation, because males, they only have one X, and females have two, so we can't express double the amount of proteins. So for female mammals, one of those X's is selected at random to be silenced. It can either be the X from the maternal side or the X from the paternal side. And that happens actually really early on in development, and that can explain how you get calico cats, right? Where you get those different colors. So the color for the orange and black color is found on the X chromosome. So during development, they randomly turned off either one or the other, and then all the cells that came from that original cell would keep the same pattern. So that's how you get like orange and black patches on calico cat. Okay, so that's constitutive. It'll be turned off forever in that female. The only places where we have facultative would be any genes that are silenced, but that can still be turned back on. So sometimes you don't need that product at a certain point, but then later you might need to turn it on. So those would be facultative. And then this one, what do you all think? So the default state for most genes, do you think that the default state is euchromatin or heterochromatin? Everybody agree? It is heterochromatin, yeah. So the default is that most things are turned off. And then if we need them, we have to induce turning them on. Okay. So how do we switch then? If you are already turned off and you want to turn on, you have to somehow tell the nucleosomes to change their shape. So how is that determined? What regulates chromatin dynamics? So the idea is that there's actually a code. It's called the histone code hypothesis. So this is this idea that if you remember those histones, and we said they have tails. The tails can be extended out, or they can be kind of tucked in, but the tails themselves get modified. So they carry the code of whether they should be turned on or off. And the code is actually very, very complicated. So for this class, we're gonna kind of look at generalities. So generally, which modifications will signal heterochromatin and which modifications will signal euchromatin. But I just want you to be aware that you can have, so if we say that methylation is found with heterochromatin, there are situations where there could be methylation in an area of euchromatin, but this is just in general. So when we say right here on this chart, modification marks, we're talking about these modifications that happen on the histone tails. So for the most part, euchromatin has acetylation. So acetyl group, which is a 2-carbon group, gets added to those histone tails. For heterochromatin, for the most part, it's associated with methyl groups that are added to the histone tails. So it's mostly associated with methylation. You can see that in the picture. So that area that's packed tightly has a lot of methylation and less acetylation, and then euchromatin has mostly acetylation, and in the picture you see that it's like the 10 nanometer fiber. So let's talk about these modifications a little bit. The first one is acetylation. So how does this actually work? Acetylation is going to cause heterochromatin, which is tightly packed, to unravel and open up. And the way that this is thought to happen is sort of a static charge hypothesis. So I have this demonstration. The groups that get acetylated are lysines. And lysine, does anybody remember what type of amino acid lysine is? Or if it has a charge? It does have a charge, yeah. So is it positive or negative? Positive. So it's one of those basic residues. So I have these lysines here. So if you imagine, like, if my body was the nucleosome itself and my arms were the tails, on the tails there's a whole bunch of lysines. So I'm going to attach them all like this. I need a rope. I didn't bring a rope for myself for this demonstration, but okay, so there's a whole bunch of lysines that are on the tail. So if we think about the DNA's wrapped around me, would the lysines be open, or would they be attracted to the negatively charged DNA? They'd be attracted. So all these positive charges, they would be wrapping around. So the tails would be wrapped. If the tails are wrapped, they're able to pack tighter, right? So when we add the acetyl groups, here's what the lysine looks like with no modification on the left. It has the charge. So the charge is attracted, it's all wrapped tightly. If I add an acetyl group to the lysine, what does that do to the positive charge? Yeah, and not only that, but it also has a carbonyl, and so that's negative oxygen, right? So it's sort of more repulsion. So if I sort of add acetyl groups to all these, it sort of cancels them out. It neutralizes them. So that is going to go from being closed to open when you add acetyl groups. Does that make sense? So you can remember that demonstration, but the idea is that when lysine is not modified, it's wrapped tightly because it has the attraction for the negatively charged DNA. When you add an acetyl group to that, you neutralize that, and so you then expose the tails out, and that's going to cause it to be open. I remember acetylation active. They both start with AC, so that can help you to remember that acetylation is usually found with euchromatin, and it's active. Okay, you do also need to know the enzymes that do this back and forth, because you have to somehow add the acetyl group or take the acetyl group off to get back and forth between heterochromatin and euchromatin. And the names of those enzymes are right here. So HATS stands for histone acetyltransferases. In the picture here they have CATS, but that's lysine acetyltransferases. It's actually the same thing. We'll use this terminology, HATS. So if you add HATS to cells, would you expect them to have higher expression or lower expression? Higher. Everybody agree with that? So you add HATS, that means means you're gonna add acetyl groups, you're gonna open it up, it's gonna be higher expression. The opposite is HDACs, histone deacetylases. They take off the acetyl group. So again, if you add HDACs to cells, you're gonna get it to go from open to closed. And I think this picture also shows that here. When you add acetyl groups with the hats, you get open, and now it's a 10 nanometer fiber. When you have an HDAC that removes, you go back to being condensed. Now you have a 30 nanometer fiber. Okay. Let me see it. Okay, so let's look at the last thing. So histones, I actually should add another slide because I kind of jumped straight from histone modifications and then talk about DNA methylation. So I want to make a note here that histones themselves, so the histone tails, they can be acetylated or they can be methylated. And the histone tails are usually modified on the lysine residues. So this is usually the thing that's being modified are lysines. They can either be acetylated or sometimes those lysines are also methylated. So there's sort of a competition for whether there's an acetyl group or a methylation group. In addition to histones being modified, DNA can also be modified, but it can only be methylated. It doesn't get acetylated. So for methylation, it happens both on the histone proteins, but it also happens on the DNA. And in both cases, those both lead to the silencing. So in general, methylation is causing silencing. So this next slide, I skipped over histone tails being methylated, and now just talk directly about DNA itself being methylated. Both of those things are going to contribute to the formation of heterochromatin. But in DNA, when DNA gets methylated, it only happens on cytosines. So we look at the structure of cytosine. The position that gets methylated is right here. This is the 5th carbon. So it's called 5-methylcytosine once it gets methylated. And what happens is the DNA itself gets methylated, and then it recruits proteins that start methylating the histones. So it's like a call signal for more methylation, which is then going to turn it into heterochromatin instead of euchromatin. But again, that's going to lead to silencing. So for this right here, for euchromatin, you should have acetylation. You might also want to add in that box, just for your own studying, that HACs add acetyl groups and HDACs remove acetyl groups.
For the modification one?
Yeah, I mean, it's just an extra note. It's not really what it's asking for. But. And then for heterochromatin, you should say that the histone tails can also be methylated, and that is going to lead to heterochromatin formation. Yeah.
How does the method lead to silencing? Does it like cause it to be more tightly compact?
Yeah, so that's a great question because it's not really consistent with that bulk static attraction model. It just leads to these proteins that come and reorganize the nucleosomes in a tight packed way. Yeah. Okay. So now in your heads, you're thinking DNA methylation is usually associated with silencing, and acetylation is associated with activation. I have a nice demo that I found to kind of show this. This is on, I can put the link up on Canvas too if you want to come back to it. But this is a Learn Genetics site. So this is, they have a nice website here, but this is an interactive control. And so this is kind of showing you what's happening at all different levels. So over here, it's showing you how the DNA is packed. The little tails are shown here, so you can see if they have green, they have a methyl tag. If they have red, they have an acetyl tag on them. And in this case, what would you say, what format is this single string, what would that be called? What level of packing, I guess? So it's the 10 nanometer fiber, so would that be,
Oh, then it's,
yeah, so this is just, you can see a single bead on a string, so that would just be
oh.
euchromatin, right? And so then it shows you also corresponding to that, if the DNA looks like this, the GFP gene looks like that, this is how much RNA would be produced. This is how much of the protein, the green fluorescent protein would be produced. And if you visualize the cell, how bright the cell would be. But with this interactive control, you can turn on this as that was totally open. I guess that was part of the way closing off. But if it's totally open, what are the marks that you see?
More red.
Yeah, you see more acetyl groups. And then as I turn up, it's wrapping tighter, and then also look what's happening to the mRNA and the protein, and then the whole cell. So now it's showing, okay, I'm not getting a lot of transcription, and the cell itself would be not expressing green fluorescent protein. And if you play with this, you can turn on the sound. It's really annoying. See if I have. It makes like this buzzing sound. So anyway, you can look at like as you turn it up or as you, so as you increase, makes more sound. And then as you silence it, it silences the expression. Okay. So let's go back now. All right, so we talked about methylation. To kind of show you how this is related, gene expression is really tightly interwoven with cancer, so I always like to make sort of those connections. A lot of times we talk about cancer as being, a disease caused by mutations, by genetic changes, but also there's a whole field of epigenetics, right? Has anyone heard that word before? Epigenetics?
Yeah, I don't know what it means.
Yeah, so epigenetics is not the gene sequence, but it has to do with the modifications of the chromatin that determine whether or not genes are turned on. So not only can you mutate genes, but you could also have problems with how they turn on or off, and that can also be a cause of cancer. So not only just changing them, but like controlling how much they're turned on. So when we look at most genes, I'll just make this note here about the CPG islands. Most eukaryotic genes have regions in the promoter where there's a whole bunch of C's and G's. So the CP gene means it's a C with a phosphate group right next to a G. In this little diagram, when they're shown as open circles, it means that those cytosines are not methylated. When they're closed, it means they are methylated. Well, it turns out that if you go to the promoter of a gene and you methylate those C's and the CPGs, the cytosines, you can turn off that gene. So this is a big way that gene expression is controlled. In cancer cells, they often will mess with this in both directions. So here's the example with the CPG island. This is a tumor suppressor gene, so you're supposed to have this on, but if it gets hypomethylated or hypermethylated, you're going to silence a tumor suppressor gene. So that could be one of the causes of cancer, right? If you're not expressing your tumor suppressor gene, this is not a mutation. It would be hard to find that. It just means that in those cells it's not expressing it, you have transcriptional repression. The other things that can happen with methylation in cancer is, we said that centromeres always are heterochromatin, right? Well, in some cancer cells, they cause hypomethylation, which is going to turn those on, and it leads to this mitotic recombination and instability that also can lead to cancer. So with cancer, like even if you can do genome sequencing on somebody, sometimes you can't actually find the mutation. Sometimes it's actually in the epigenetics, in the way that cells are expressed, whether they're turned on or off. So here's that, that definition. The epigenome is a collection of all of those chemical modifications, the acetylation, methylation. There's other modifications too. You can see phosphates, phosphorylation. So those are the examples that can be part of the epigenome. And I have a nice, if you're interested, well, this is a very short, let's just watch this really quickly. What is, oh, sorry. And, okay, what is the epigenome? As long as it has closed captioning, because I don't think I can hook up my,
DNA contains the instructions for building all the parts of the body. But DNA is only half the story. The DNA in our bodies is wrapped around proteins called histones. Both the DNA and histones are covered with chemical tags. This second layer of structure is called the epigenome. The epigenome shapes the physical structure of the genome. It tightly wraps inactive genes, making them unreadable. It relaxes active genes, making them easily accessible. Different sets of genes are active in different cell types. The DNA code remains fixed for life, but the epigenome is flexible. Epigenetic tags react to signals from the outside world, such as diet and stress. The epigenome adjusts specific genes in our genomic landscape in response to our rapidly changing environment.
okay. So have you all heard of nature versus nurture? That's always a conversation about, what controls our traits, whether it's our environment or whether it's our genes. Well, the epigenome is part of sort of the nurture aspect, the environmental effect. So as it said, your diet can affect epigenome, stress, and that can actually be multi-generational. So you can have, they've done studies where they've looked at, a fetus that is inside of its mother, and she experiences a lot of stress, and the cortisol affects the epigenome of that child, which can have later impacts for, their risks of diseases or, like diabetes. They've looked at all these different things. So it's a very complicated thing, and it's kind of hard to track down. And I guess it's an interplay between your genome and your epigenome. One of the other really interesting things is that, how many of you know identical twins or have known identical twins? So for identical twins, anybody know how they arise versus fraternal twins? Yeah.
So identical twins is one embryo that gets split in two, whereas fraternal twins are two eggs and two sperm that are created.
Yeah, exactly. So identical twins, in theory, have the same DNA, right? But if you've known identical twins, you know that they do have differences. Sometimes you can visually tell them apart by differences in the twins. And that actually can help to explain, or that can be explained a lot by epigenetics of twins. So we already talked about epigenetics, which is the study of traits that's due to changes in the gene expression or changes in the epigenome. This experiment right here, they took 3-year-old identical twins and they looked at one of their chromosomes. And so in this case, they had, for one of the twins, they used a green label, and the other twin, they used a red label. They overlaid their chromosomes, and you can see that when they were 3, they had very similar marks. They had very similar epigenetic tags at that time. But then they looked at another set, I don't think this was the same set in this particular study, but they looked at a pair of 50-year-old identical twins, did the same thing where they labeled one of the twins with green, one of the twins with red, looking at that same epigenetic mark, and they found that there was very little overlap after 50 years. So as twins age, they are going to have mutations that do occur individually, right, in their somatic cells, but also a big effect is that their epigenome is going to change as they age. So those epigenomes will become more different throughout their lifespan. And that kind of explains when they do these identical twin studies, sometimes there's confounding results. They're using the twins as being like exactly identical, and in actuality, they're really not identical. All right, anybody have any questions on any of that? Okay. So I think we can do one more little segment and then we'll take a break. I want to stop at 12:45. Okay. So we're going to just briefly touch on the transcription regulation. We already, for prokaryotes, we talked about, the LAC system. We're mainly going to focus on how this is different in eukaryotes. So in eukaryotes, we don't use the operon idea. There's not this kind of clustering of genes that are working together under the control of the same set of regulatory sequences. Instead, eukaryotes can coordinate regulation of genes by regulating something else. Does anybody know what that might be? They don't control them by putting the genes together. Does anybody remember what they can control?
They control like what interons are spliced out.
So that is, they can control that. So that would be like alternative splicing, but I guess thinking about coordination, like in the liver cell, if you needed all these genes that are specific for liver cells, how could you make sure they all get turned on?
Is it specific transcription factors?
Yeah. So you could turn on specific transcription factors for specific cell types. So for this blank, we can write by regulating expression of transcription factors. And so which cells have which transcription factors basically, tells you what genes they're going to be turning on in those cells. So your neurons have neuron-specific transcription factors. Liver turns on a whole different set of genes. Okay. The other things that are sort of extra in eukaryotes, we talked about how they have a much more complicated promoter region, and specifically we talked about the core promoter. The proximal promoter was right next to the core promoter, and then they can have things that are really far away, right, the distal promoter. So with eukaryotes, it just becomes really hard to sort of figure out what is regulating each gene because of the addition of these distal promoters. And in the distal promoters, we have enhancers and insulators. So I included both of these. These are both examples of some of those DNA sequences. They can be 10,000 base pairs away, controlling a gene way far away. So this just shows you, comparing bacteria, they have their sequences right next to the promoter. With yeast, they start to have some further elements. With humans, we've got all of these sequences that are really far away. So there's been a lot of projects that have been done where people have been trying to, identify where are the enhancers. If they're up to 10,000 base pairs away, that's a big puzzle to figure out, which gene is this enhancer working on. So we did not mention insulators before. Insulators are sort of boundary elements. They're basically, if you think about the fact that enhancers are really far away, let's say that you have, I need two colors. So you have this gene right here, gene 1, and then the DNA is going over here, off to another region, and let's say they have gene 2 over here. If you had an enhancer that was right here, And you need a way to make sure that this is only acting on this gene and it's not affecting the expression of this gene. That's what these insulators do. They set up a boundary so that this enhancer cannot act on this gene. So the insulator sequence would be somewhere in here to prevent this enhancer from working on the wrong gene. So, I mean, suffice it to say that it's just very complicated how all of this is regulated together in eukaryotes. But they help to define regions so that the enhancers know which genes they're supposed to activate. Okay, so the very last part here that we will talk about is the transcription factors themselves. They usually have this kind of a structure. They have to have some region that can recognize DNA. So they have a DNA binding domain. This is the two little feet that are shown here. The DNA binding domain will look for a specific sequence. Does anybody remember which part of the DNA those proteins interact with? How did they figure out the sequence?
Yeah.
You remember it?
Outer group.
The major group,
Major
right?
group.
So they recognize through hydrogen bonding. So this blank should be major group. They interact with the major group, and that's how they find a specific sequence that they are looking for. Then they often are found as dimers. The reason for that is that this extends the sequence specificity. Because if you can only touch, like let's say you can only touch 4 bases with one part, by having a dimer, you're actually looking for a sequence that is 8 bases long, which is gonna occur at random less frequently, 8 compared to 4. So a lot of times they dimerize, they have to dimerize with each other through this domain. So this middle region is the linker domain. And then this top part is sort of the activity part of the transcription factor. This is going to facilitate or inhibit transcription. So that might have to do with helping to recruit the mediator, or it might have to do with helping to recruit euchromatin factors. So that activation domain has lots of different possibilities, but it's going to increase or sometimes will inhibit transcription. All right, so this, well, I guess it's already, this will be our last slide. So the transcription factors, this is just kind of reminding you about how they recognize the sequence by binding in the major group. And this one shows you one particular example, and you can see where it lays down is right in the major group here and on the back side. So this one is a dimer. Those would be the DNA binding domains, and then this linker domain is how they dimerize. And up here, this is where the activation domain would be. Okay, so we'll stop here. We'll finish this off tomorrow, so we'll finish off the rest of this. So let's go ahead and take a little 10-minute break. And when we come back, we can pick our worms to set up the paralysis app.